Great. All right, so this is the last bit of what we call our macronutrients. So we talked about carbohydrates, we talked about lipids, and now we're going to look at the structures of proteins. The rest of the stuff that we're going to do is more about how the body functions. So we're going to talk about a little bit about enzymes and molecular genetics, DNA and RNA. So what are proteins made of? Amino acids, yeah, very good. We've looked at amino acids a little bit before when we looked at the functional groups. But let's now take a look more specifically about their structure and how they would actually look in situ, so in a solution like in the body. Now that we know a little bit more about stereochemistry too, we can know some stuff there as well. So proteins are made from amino acids. Amino acids are small molecules that all have roughly the same basic structure with one spot of variation. So an amino acid, on one side we'll have a carboxylic acid, but now that we're being a little bit more precise and we're talking about inside biological systems, rather than make that a COOH carboxylic acid, we're actually going to make it the conjugate base of that acid, which is where it loses its hydrogen and becomes a carboxylate. So when this has lost its hydrogen, we call that a carboxylate, and we'll see why it's that form like that in a minute. On the other side of that carbon is the amino group. So you got the acid and the amino. And when we drew these before, we talked about amines. We had neutral nitrogen with two other connections. But now we're going to see that actually in these structures, it is the conjugate acid of an amine, which is ammonium. So what we call the acid is really a carboxylate, the conjugate base of a carboxylic acid. And what we call the amino is actually the ammonium, the conjugate acid of that. And the middle part, we'll talk about that in a second. The middle part is a carbon with usually a hydrogen and then some sort of R group. And this R group is what distinguishes all the different amino acids from each other. So all the amino acids have this two-carbon basis with the amino part. They all have a carboxylate or carboxylic acid, and they all differ by what's hanging off on that R group. Right. So why do we draw it as plus on the left and minus on the right? Well, this has to do with how acids and bases work. In a neutral solution, a carboxylic acid is actually more acidic than neutral, so it wants to give up its hydrogen. And an amino compound, a nitrogen, just like ammonia, is more basic than neutral, so it wants to accept a hydrogen. So if you have a molecule that has an acid side and a base side, that hydrogen is going to get transferred from the acid side to the base side. And so that's why we always see amino acids now. When we see them now, we're going to see them in this way, we're going to draw them in this way, and show that there's a positive side and negative side of the amino acid. Now, when the amino acid is in non-neutral conditions, that'll be different. So if this is in acidic conditions, then there will be an extra hydrogen that goes onto the acid. So there will actually be an overall positive charge. When we're in basic conditions, there will be an extra, or there will be a negative on the carboxylate and a neutral on the amino side. We're not going to mess with that too much. But that's one of the reasons why it's so important to control pH in different parts of the body, because it's going to change how the structure of the amino acids are. Okay, let's look at the stereochemistry of amino acids. So that middle carbon right here, this is called the alpha carbon. Alpha in chemical terms usually means like one away from something. So these are actually often called alpha amino acids. We just call them amino acids. That alpha carbon is what we call a stereocenter. It's a center of stereochemistry, which means that it's going to matter which direction those various parts are going. So we're going to have L amino acids and D amino acids that are mirrors of each other. Normally when we draw these, we use Fisher projections just like we did with the carbohydrates. So let's draw those. So here's our carboxylate up here. And we usually put the R group underneath. If the R group is a CH3, that makes this alanine. And then the stereochemistry is given by the by where the amine is in this situation. So the amino part would be over here, and the hydrogen would be over here. The amino part being on the left makes this an L amino acid, L-alanine, just like the OH in the sugar has made that an L sugar. So that means that the mirror image of this is not the same. This is D-alanine. So all amino acids isolated from nature are L. Almost all. Well, yeah, we'll say almost all. And so even though that looks like a small difference, just like we talked about with carbohydrates, the difference between that orientation in space between having a D versus an L amino acid is going to make a huge difference in how it interacts with your body. You cannot, you have to eat a bunch of proteins to do stuff, to have your body do stuff. But if that ends up being D proteins, it's not going to work. And it's probably going to be bad for you. At best, a little bad for you. Or I guess at best, you just won't use it. At worst, it'll hurt you. The most natural amino acids are L. I'm going to show you a table from your book of amino acids. You don't have to memorize names of amino acids. But if you go on and do any kind of biochemistry or like medicinal stuff or physiology, you may have to memorize them at some point. So let me copy this image, and we'll put it over in the notes to look at. So each amino acid has this form, as we talked about, with the R groups, and they all differ from these R groups. So if you've heard of some of these different names, you can see how the different amino acids have these different R groups. So you've got phenylalanine, alanine, glycine, tryptophan, methionine, all these different ones. They also generally will have a 3-letter abbreviation because we have to think about how they hook together, and we usually make like these chains of them. And they also often have a 1-letter abbreviation, which isn't given here, but is given in another table. Because when we're talking about sequencing proteins, and we want to know like what is the amino acid sequence of this whole protein. You're talking about hundreds or thousands of amino acids. And if we want to know that sequence, we use just single letters to kind of spell it out. So then we know what's connected to what and what's connected to what. All right. So you don't need to remember these structures, but what you should be able to do is group them based on our groups, and not by memorizing anything, but just by thinking about what we've already learned. So these are often categorized in this way, where you have the hydrophobic R groups, that is the lipophilic or the non-polar R groups. And so you'll notice all of the R's in this category can pretty easily be seen as non-polar. All carbon, hydrogen stuff, no oxygens or nitrogens in sight, with the exception of this one here, which is tryptophan. But you can see there's really no source of polarity in those structures. All right, so then you have the polar amino acids here, and you can see how each of their R groups have either hydrogen bonding, OHs and NHs, or this SH here and cysteine. So those are our polar neutral R groups. And then we have some groups that are charged. So you've got your positively and negatively charged R groups as well. And you can imagine how those might interact differently. And as we'll see as we look at the larger structures of proteins, those end up having a big effect on how the protein organizes itself. All right, so no need to memorize that stuff, but based on what we've already done, you should be able to determine if R groups are polar or non-polar. All right. So to look at the primary structure of proteins, yeah?
So I noticed that alanine, it's H3.
Okay. All right, so we're going to look at a couple levels of protein structure, and I don't think we'll get to the highest level today, but we'll look at a couple of the levels. The first level is just how these amino acids hook together to make proteins, and that's really important. It's something we've seen already, and it's called a peptide bond. So sugars hook together through glycoside bonds. Fats we talked about hooking together through glycerides, and now we're going to look at proteins and peptides. So to make a peptide bond, you take two amino acids and you lose water the same way that we did with the glyceride bonds last week. Or, no, it was just Tuesday, right? Yeah, Tuesday. So let's take a look at how these might connect. So I'm going to look at a molecule of glycine. Glycine has just an H for an R group. And we're going to take a look at, and then there's its carboxylate. And then we're going to react glycine together with alanine. I'm going to draw this one in blue so we can see it. And I'm gonna draw all the hydrogens out on the nitrogen just so we can see, because they end up doing stuff in this particular example. So now instead of an H for an R group, we have a CH3 for an R group for alanine. And then it's carboxylate over here.
Connected to the oxygen? The H and the oxygen.
These are not, well, this is connected, that's its own molecule. So that's its amino acid. So here's glycine. And here's alanine. So just as with the glycerides, we're going to dehydrate, so we're going to lose water. And that ends up connecting the nitrogen to the carbon here. So everything else stays the same. But now, whoops, How many hydrogens? Now they're connected together through this peptide bond. It still has a positive amine side on the left, still has a negative carboxylate side on the right, but now it has this new connection in the middle, and then water is the byproduct. So this new connection here is called a peptide bond. And we also know it as something else. What's the other name for that functional group? Not quite. Aldehyde would have a hydrogen on one side, connected to the carbon with the carbonyl. Yeah, that's an amide or an amide. That functional group is the same, but when it's amino acids is the only time we call it a peptide bond. All right, so now we've made a link between two peptides. So we've made a dipeptide, or between two amino acids, so we've made a dipeptide. This particular dipeptide is going to be called, or it's going to be named GLY-ALA, because those are the abbreviations for glycine and alanine. And the order is important because we could have also put alanine on the left and glycine on the right. That would have made a different dipeptide. So GLY-ALA is this specific one.
Is the whole structure
Mhm.
turned into?
Yeah, that whole structure. And then we could keep hooking things on to both ends to make even longer peptides and make polypeptides. And so I don't want to draw these big ones because they get a little crazy, but let's take a look at what a slightly larger one might look like. Don't worry about that first one. But here is a polypeptide of 1, 2, 3, 4, 5, 6, 7, 8 amino acids. And you can see that along the middle, red here is oxygen, and green is nitrogen, and black is carbon, and the little white ones are hydrogen. So you can see how in 3 dimensions, you end up getting this kind of chain where all the peptide linkages are. And then the R groups end up coming off the chain, kind of alternating up and down as it goes. And so we could have constructed this in the same way, drawing it step by step, one after the other. And this is called the primary structure of amino acids. The primary structure defines how the amino acids are hooked together. So like what order are they hooked in, which one is connected to which. And so one thing I might ask you to do on the exam is draw a polypeptide of given amino acids. And I would give you the structures of the amino acids, but you would have to put them together, peptide bonds.
So on all 3 of those, is that a negative sign next to the oxygen, or is that similar to hydrogen?
That is a negative sign. So that oxygen has a negative charge because it's hydrogen.
Is it the same protein?
What?
It won't have like a negative charge in proteins.
That's right. So proteins will always have a negative charge side and a positive charge side. The oxygen, the carboxylate side is negative, and the amino side is positive. We'll see that in larger structures.
Then wouldn't that not be a carboxylate?
Right. So it's technically a carboxylate instead of a carboxylate. This form. All right, so let's look at the secondary structure. The secondary structure is, okay, we know how the amino acids are put together, but then what happens in 3 dimensions when we have that putting together of amino acids? And what happens turns out to be relatively predictable based on the structures of these amino acids. And there's basically there are 2 main types of secondary structure. There's what we call an alpha helix, which I'm not going to draw, but we can look at the picture in the book. So this is a chain of amino acids. And once it gets long enough, rather than stay straight, it starts to twist. And the reason it starts to twist is because if it twists in this helix, then suddenly the hydrogens on the nitrogen and the oxygen from the carbonyl group are close enough to hydrogen bond. So those hydrogen bonding interactions we talked about a few chapters ago that we said were really strong and stabilizing and important, that ends up controlling this whole structure. So instead of stretching out, they kind of twist so they can be a little bit closer. And these hydrogen bonds hold this together in a pretty rigid way. So this ends up actually being a pretty, even though we say that hydrogen bonds are weaker than regular covalent bonds, that ends up being a pretty strong interaction when you take them all together. And this ends up being a pretty solid part of what makes up a protein. So that's an alpha helix. I can put that image here, but I'm not going to draw it. If you look kind of down it, you can see that all of the nitrogens kind of end up in the inside here. And then the hydrogens and the hydrogens aren't shown in this picture, but they're out so they can connect to the oxygens. And it makes kind of this tube-like structure. The other main component of the secondary structure is called a beta sheet. And this is another way that amino acids chains can arrange themselves to maximize hydrogen bonding. So first of all, when we draw protein structures, I guess we should say this. Alpha helix, we just draw as like this, as you can see. And a beta sheet, we usually draw like this. An arrow, and then that'll be connected to other stuff. But the arrow indicates where the nitrogen is and where the carboxylate is. So the flat end of the arrow is the ammonium part, and the pointy end of the arrow is the carboxylate part of the chain. And beta sheets can stack together and hydrogen bond as well. Well, it can go the whole other direction, but the pointy end is always the carboxylate, and the flat end is always the amino part. Let me show you a picture. So, where's that picture? Here. No, it's not. Not here. Okay, let's try. So here are some beta sheets. So you can see the carboxylate end is always on the pointy end, and they can line up kind of parallel and interact that way, or they can line up anti-parallel like this. If we zoom into the amino acid level, kind of like we did with the helices, here are some glycine-alanine alternating peptides. So you can see here the polypeptide goes alanine, glycine, alanine, glycine, alanine, glycine, alanine, glycines. That way they can line up in this particular way where they maximize these hydrogen bonding interactions inside the sheets. And there you've got your beta sheets. Yeah, and
Is there a more common one, or which one's stronger?
they're both components of most proteins. So most proteins will have alpha helix pieces and beta sheet pieces in addition to other stuff. major structural components of those two things. I don't know if you can say one is necessarily stronger than. So the last thing we're going to look at is called the tertiary structure. So you take these alpha helices and these beta sheets and whatever, and all the other stuff going on the active site, if it's an enzyme and whatever else, and then this protein, even zoom out more than that, starts folding on itself and connecting in different ways. And so the way that these proteins fold actually ends up being just as important, if not more important, than what they're actually made of. So we're used to in chemistry of only caring about like the smallest parts, what's bonded to what and all of that. But in a protein, this macro structure, what the whole thing actually looks like, which is kind of like that, is just as important for how it works in your body or in whatever system it's in. So there are essentially 4 types of interactions that control a protein's tertiary structure. You have what we call hydrophobic interactions, which are nonpolar attractions, dispersion forces, things from like aromatic rings attracting each other. You have hydrogen bonding, which we've talked a lot about. So nitrogen, hydrogens, and carbonyl oxygens can connect together and make hydrogen bonds. You have what's called a salt bridge, which is positive and negative components. So the positive ammonium component attracting the negative carboxylate component. And then you have what's called disulfide bridges, which are covalent bonds, but they're not particularly strong, meaning I guess they're strong with their labile, they can break easily. And those also kind of hold things together. So when you put all that together, you get this very specific thing. And what we did in lab on Tuesday and a couple weeks ago with the milk too, is that we broke all these things apart. So when you add things like ethanol or acid or heat, you break apart these interactions, which causes the protein to spread out, and then it gets tangled with itself, and it reforms in a different way. So I think there's a picture of this. Somewhere. There's that picture. Here, 653. So we'll get through more of this after the exam. I want to spend some time to go through what the exam is going to be on. But this is basically what we were doing in those labs. You took your proteins, which in this example are alpha helices, but they could be whatever. Then we denatured them, so we broke those hydrogen bonding interactions or those salt bridges or whatever, spread them all out, and then they coagulate and form those chunks. And that's how you form like the cheesy curds in the milk, and that's how you form the white precipitate in the albumin on Tuesday, and all of that. That's called denaturation. So that's breaking, even though the amino acid chain is still intact, you're breaking all these different intermolecular force type interactions, and then it reforms in this new way. So we'll look at that more after the exam when we get into some of the function of proteins and talk about why they have to do that and why they have to be their specific shapes that they have to be. All right, so let's take a look at the objectives, and I think if you have any questions, we'll go through that also. Okay, so as you saw with the last exam, I'm not going to ask you like really specific random questions from a million chapters ago, but the test should be considered to be cumulative because all of those skills from before are still important now. So things like understanding hydrogen bonding versus dispersion forces, and what's polar and what's not polar, and how chemical structures work, and how to figure out if you have like how many hydrogens you have versus how many carbons you have in a line structure. We've been dealing with a lot of line structures now. If you're still sort of just nodding your head and not really knowing what that means chemically, make sure you brush up on all that stuff because that's going to be big. So always before an exam in this class, it's good to look over all of the objectives going back and highlight some of the ones that you know you don't quite have or you never quite learned in the first place, and give yourself some time to catch up on those. Things like naming compounds, drawing reactions, especially the intermolecular forces stuff, because that's so important with the biomolecules and the organic chemistry.
Yeah, is that on a test, being able to draw line structure, or was that the last test?
Both, yeah. Okay, so with the new stuff, so I guess the new stuff would be the functional groups. I just created that quiz, and it was pretty clear who got those and who didn't. So if you still haven't memorized the functional groups, what they're called and what they look like, and how to distinguish them from each other, please make sure that you do that. In the biomolecule stuff, it's pretty much what you saw in the quizzes. So identifying the sugars, looking at the three-dimensionality of what are enantiomers, what are stereoisomers. I'm not worried so much about those definitions, but what I want you to be able to do is determine whether things are the same or whether they're stereoisomers. I think that's what's really important. Can you draw the carbohydrates to determine D and L from those structures, like was on the quiz? And then the hydrolysis and dehydrolysis, we didn't really, I didn't put that on the quiz, but what we talked about that was with the glycoside formation. So that's what hydrolysis and dehydrolysis is. When you make glycosides, how do you hook sugars together? What do they look like when they're hooked together? And how would they then break apart? Kind of like what we saw on the quiz. All right, for the lipids, which we haven't had a chance to do a quiz on because we just did that on Tuesday, make sure you understand those terms, specifically unsaturated versus saturated, triglycerides, diglycerides, monoglycerides. What does that mean? How do those reactions work? So hydrolysis reactions and dehydrolysis. Dehydrolysis is the glycoside, or is the glyceride formation. And the hydrolysis is what we did in the lab with soap. And we said we could also do that with acid. And then we talked a little bit about the phospholipid membranes and the phospholipid lipoproteins that you see in solution. So you should be prepared to discuss those things. And particularly, as you can maybe guess, I'm most interested in understanding the intermolecular forces, why they do what they do, and explaining that. So I'm not going to ask you to probably draw a phospholipid bilayer membrane, but I may ask you to describe why it orients the way it does. Why do we need the phosphate parts on the outside and the lipid parts then go to the inside? Those kinds of ideas, which again all comes down to intermolecular forces, polar versus nonpolar, which we talked about chromatizing. And then of the new stuff for proteins, I mean, you can probably do all of this, but not this stuff. We'll get into that later. I would say for proteins, focus on amino acid structures and the stereochemistry that we talked about, and condensing amino acids into peptide chains. So what does it look like when amino acids come together to form peptide chains, and how would you draw that? You don't have to memorize amino acid structures. Probably what I'll do is just give you a table much like the one that we just looked at that has all of the amino acids on it with all their names. And so when I say in a problem like draw a peptide between glycine, alanine, and leucine, you can look up those structures and figure out how they're going to go together. So, Jen, your question.
Yeah. We went over it really briefly, but what about steroids? Do we know anything about that besides the structure?
Yeah, I would say know that steroids are made from that 4-ring structural feature. You know, the like 3 5-membered ring, 6-membered rings, and 5-membered ring. So you should be able to pick out like here's this steroid. Okay. I would guess that I haven't written it yet, but there's probably going to be some sort of a matching component where like, you know, it'll say monosaccharide and steroid and whatever, and you have to say which one is which. No. It's on there, isn't it? Yeah, I don't think I would test you on that for this exam. You may want to brush up on it for the final, but I don't think it's on this exam. Yes, it says objectives. The names of the alkanes. That was technically last exam stuff, even though I don't even know if I put it on your last exam, but there's enough.
Wait, so some of the functions run on.
No, know the functional groups, but the names of all the alkanes like methane, ethane, propane, butane, pentane, all those. Other questions?
So we don't have to draw anything, or it's sorry.
Yeah, you got dressed up.
So draw structures.
Yeah, definitely. I think what I said specifically was
Are we gonna have to draw it on the test? Or gonna have us connect them?
both. Yeah, you should be able to draw the. I mean, you don't have to memorize the structures of the amino acids, but if I give you the structures of the amino acids, you should be able to draw peptide chains based on how they connect to each other. Drawing those peptide bonds.
That's just,
Yeah.
we have to describe some different anatomy.
No, we haven't got to that yet. No, I would say we stopped at like 5-ish. Yeah, so whatever the end of that page. Whatever the end of that page. Yeah. Functional groups and we'll do more stuff with structures too.
So the peptide is just the loss of the water and just kind of like right regardless of.
That's it. All right, send me messages if you need anything this weekend. I'll be getting grades updated. And yeah, otherwise I'll see you for the exam on Tuesday. We'll do our usual schedule on Tuesday, so we'll kind of start with a review or whatever, and then whenever everybody's ready, we'll start the exam.
All my quizzes.
Okay, what's your grade with all the zeros?
Well,
The classes are not coordinated anymore.
Yeah, this way I have another
And
class.
so they have the exam after next year. So it'll actually be a different, it'll be different. Material. It doesn't, whatever.
Like a what?
Yeah.
Is that?
It's because they're
No, it's just not part of the floor.
through a folder. Your blood is mostly full. So the outsides are phosphate bonds are
So you have a little bit of
water. So they can carry those down water.
I think I got, they have the first page, right? Definitely got this one wrong.
Triglycerides.
I got it all right.
would not dissolve in water.
Oh, it would.
But inside those, but
No, Lord, I want to see.
polar versus non-polar. Same thing with the bilayers. You've got the phosphate inside, whereas
Okay.
inside the cell is water, and outside the
You
cell
want
is
this more?
But
Picture little.
I hate you.
barrier between
Little.
between those two
Tomorrow, what's tomorrow?
cells. And it's very
What time do you want to be here? I'm here for. Compare दो बजे. Compare दो. You want to do noon?
The book shows that.
Or Monday?
Oh yeah, because
Monday I'll be sleeping.
Yeah, I can back to work at like.
I have a question.
Yeah.
If